are called disaccharides. For example, sucrose yields alpha-D-glucose and beta-D-fructose on hydrolysis. This means that disaccharides are formed by condensation reaction between two monosaccharide units. Hemiacetals of monosaccharides undergo condensation between their OH groups with the elimination of water molecule. The bond which holds the monosaccharide units together is called glycoside linkage. All disaccharides are crystalline solid, soluble in water, and fall into two classes. Reducing sugars. If the reducing center of any monosaccharide unit is free, the disaccharide is reducing. And non-reducing sugars. If the two monosaccharide units are linked through their respective carbonyl groups, the disaccharide would be non-reducing. Sucrose. Sucrose is the most widely occurring disaccharide and is found in all photosynthetic plants. It is white crystalline solid and soluble in water. When heated above its melting point, it forms a brown substance called caramel. It is commercially obtained from sugarcane and sugar beets. When sucrose is hydrolyzed by dilute acids or enzyme invertase, it gives an equimolar mixture of D-glucose and D-fructose. Sucrose is dextrorotatory with specific rotation of plus 66.5 degrees. On hydrolysis, sucrose gives an equimolar mixture of dextrorotatory glucose and levorotatory fructose. The conversion of sucrose into equimolar mixture of glucose and fructose is accompanied by change in sign of optical rotation. Therefore, hydrolysis of sucrose is called inversion of sugar. The equimolar mixture of glucose and fructose is called invert sugar. Maltose. When starch is hydrolyzed by the enzyme diastase, maltose is formed as one of the products. On hydrolysis with dilute acids, 1 mole of maltose yields 2 moles of D-glucose. Maltose is a reducing sugar. In maltose, the two D-glucose units are linked through alpha-glycosidic linkage between C1 of one glucose unit and C4 of the other. It tastes sweet and it is not found freely in nature. It has the ability to reduce the Fehling solution due to its free aldehyde. The aldehyde group is oxidized, giving a positive result, which means that the maltose is soluble in water. In aqueous solution, it exhibits mutarotation.
Lactose. Lactose is a disaccharide sugar derived from galactose and glucose. Lactose crystals have a characteristic tomahawk shape that can be observed with a light microscope. It is found mostly notably in cow milk. It makes up around 2 to 8% of milk. It is used in the pharmaceutical industry. It is added to pills as a filler because of its physical properties and low price.
Polysaccharides. Polysaccharides are polymers of monosaccharides joined together by glycosidic linkage. These are known as glycans. Polysaccharides that are polymers of a single monosaccharide are called homopolysaccharides. A homopolysaccharide consisting of glucose monomeric units is called a glucan. 3 important polysaccharides in which all are glucans: starch, glycogen, and cellulose. They are polymers of glucose and differ from each other in molecular weight, the nature of linkage between glucose molecules, and degree of polymer chain branching. Starch. It is a white amorphous powder sparingly soluble in water. When heated with water, it produces a colloidal suspension from which two major components can be isolated. One component is called amylose and the other amylopectin. Amylose. It is soluble in water. It is a linear polymer of alpha-D-glucose. And amylose consists of more than 1000 D-glucopyranoside units, which are connected in alpha-glucosidic linkage between C1 of one unit and C4 of the next unit. Amylopectin. It is water-soluble fraction of starch. Branching takes place between C6 of one glucose unit and C1 of another, and occurs at intervals of 20 to 25 glucose units. It has a molecular weight of 1 to 6 million. Cellulose. Cellulose is a linear polymer of D-glucose units joined by beta-glycosidic linkage. The chains are held together by hydrogen bonds between glucose units of adjacent strands. The arrangement of chains constitute the bundles. This tends rigidity to its structure. Cellulose is also formed by condensation of D-glucose units. It does not reduce Tollens reagent or Fellings solution. It is not fermented by yeast. It cannot be hydrolyzed easily. When it is heated with dilute sulfuric acid under pressure, it undergoes hydrolysis and yields D-glucose. Glycogen. Glycogen is a multi-branched polysaccharide that serves as a form of energy storage in animals and fungi. In humans, it is made and stored primarily in the cells of the liver and the muscles, and functions as the secondary long-term energy storage. It is a polymer of glucose with 1,4-alpha-glucosidic linkage and having considerable chain branching. It is white powder, soluble in water. It gives purple color with iodine. Molecular weight of glycogen is very high and ranges from 2 into 10 raised to the power 5 to 10 raised to the power 8. It is found in nearly all animal tissues. Liver is the principal site in which it is stored. When body needs glucose for energy, enzymes break glycogen down to glucose. Importance of carbohydrates. They act as biofuels to provide energy for functioning of living organism. They act as constituents of cell membrane. It helps in breakdown of fatty acids and preventing ketosis. Derivatives of cellulose are used in explosives, lacquers, celluloid, and collodion. It helps in regulation of various metabolic processes in the body. Cellulose acetate is used in photographic film and packing materials. It is present in storage form of food in plants as starch. Proteins. Proteins are the most abundant biomolecules of the living system. Chief sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. They occur in every part of the body and form the fundamental basis of structure and functions of life. They are also required for growth and maintenance of body. Proteins perform a vast array of functions within living organisms, including catalyzing metabolic reactions, replicating DNA, responding to stimuli, and transporting molecules from one location to another. Amino acids. These are amino-substituted carboxylic acids. The alpha amino acids are the building blocks of peptides and proteins. In alpha amino acids, the amino group is present on the alpha carbon atom. Amino acids are classified as neutral, acidic, or basic depending upon the relative number of amino and carboxyl groups in the molecule. Neutral amino acid. If the number of amino groups and carboxyl groups is equal in the molecule, the amino acid is neutral. For example, glycine. Basic amino acid. If the number of amino groups is more than carboxyl groups, then the amino acid is basic. For example, lysine. Acidic amino acid. If the number of amino groups is less than carboxyl groups, then the amino acid is acidic. For example, aspartate. Essential and non-essential amino acid. Amino acids can be synthesized by all living organisms, plants, and animals. The amino acids which can be synthesized in the body are known as non-essential amino acids. Many animals are not able to synthesize all the amino acids they need for their proteins. Those amino acids which cannot be synthesized in the body and must be obtained through diet are called essential amino acids. For an adult human, there are 10 essential amino acids. Deficiency of essential amino acids in the diet may cause diseases such as kwashiorkor. Peptide, polypeptide, and proteins. Enzymes can cause alpha amino acids to polymerize to give peptides through elimination of water molecule. The CONH linkage that forms between the amino acids is known as a peptide bond or peptide linkage. Amino acids, when joined in this manner, are called amino acid residues. The polymers that contain many amino acid residues are called polypeptides. Polypeptides are linear polymers. One end of a polypeptide chain terminates in an amino acid residue that has an NH2 group is known as N-terminal. The other terminates in an amino acid residue with COOH group is known as C-terminal. Proteins are molecules that contain one or more polypeptide chains and have molecular mass higher than 10,000. The precise order of bonding in a peptide is called its amino acid sequence. The amino acid sequence is specified by using 3-letter amino acid abbreviations for the respective amino acids and connected by hyphens. Classification of proteins. Proteins can be classified into 2 broad classes on the basis of their structure: fibrous proteins and globular proteins. Fibrous proteins. When polypeptide chains run parallel and are held together side by side at many points by hydrogen and disulfide bonds, a thread-like structure is formed. Such proteins are called fibrous proteins. They are insoluble in water. They serve as the chief structural material of animal tissues. For example, keratin in skin, hair, nails, horn, and wool. Globular proteins. The polypeptide chain in these proteins is folded around in such a manner so as to give the protein molecule almost spheroidal shape. They have weak intermolecular forces. They are soluble in water. They catalyze biological reactions, for example, insulin and thyroglobin. They act as regulators of metabolic processes. For example, maintains blood sugar level. They act as antibodies and protect the body from diseases. Structures of proteins. The structures of proteins are quite complex. There are 4 levels at which the structure of proteins can be described: primary, secondary, tertiary, and quaternary structures. Primary structure.